He like taking things. I know.
All right, sign sheets going around, so sign on in. The digestive system, especially after Thanksgiving, understand how everything was functioning as we were eating. Okay, so the whole goal of our digestive tract is to break down the large food stuffs made of all the macromolecules that it's made of into small molecules that we can then absorb. So we can break the food down into the very small building blocks. So that So after we have digested all of our food stuff, all of that, those small nutrients are now in our blood. It's absorbed out of the, what region of the small intestine, or what region of the digestive tract?
Yeah, me be put on top of
The small intestine, we've done all that absorption and all the small molecules into the bloodstream, right? The blood is then gonna enter into the kidneys. Enter the kidneys. And there we're gonna do some filtering of blood. And while we are filtering the blood, we're going to possibly pull out some of the nutrients that we want to hurry up and put right back into the blood. And so we're going to see how the blood makes its way through the kidneys and gets filtered. And this is how we regulate our water volume in the blood. We want to maintain a volume of blood kind of constant so that we don't disrupt blood pressure. And the more watery the blood, the more volume, the higher the blood pressure, the more liquid pushing against the blood vessels. So we want to make sure that our water volume and the amount of things dissolved in the blood, volume, concentration, all that remains constant, and that hemostatic level. Here are the kidneys, the urinary system. We are getting rid of wastes, okay? We're eliminating any matter, organic matter that we want to get rid of, material that we want, nutrients. The urinary system helps to regulate blood pressure. See how the urinary system is sensing blood pressure, so that it can make adjustments. So all of our pH, the pH of the blood is also being sensed. The salt levels in the blood is being sensed in the urinary system, so that the urinary system knows that it needs to pull more salt out or leave the salt in. It's sensing what's in the blood as it's traveling through the urinary system and being filtered. And then that filtrate is what we excrete in the urine after the blood is filtered. So at the kidneys, all of our liquid part of the blood, okay, the plasma, gets filtered 60 times a day. And we call that the filtrate. Okay, the filtrate is the liquid part of the blood, no blood cells, no important blood protein, but just the liquid part of the blood. And that filtrate goes through a process. It's traveling through the urinary system. And that process is turning that filtrate into what we call urine. By the time it's become urine, it's only about less than 1% of what the filtrate was. So this filtrate goes through a lot of changes as it's traveling through the kidneys, right? The major organs of our urinary system. So much so that by the time it becomes urine, it no longer looks like the original filtrate that filtered out of the blood. Barely looks like. And so our urine is mostly water with some solutes in it, lots of nitrogenous products, urea, which is the major component of urine. We have other solutes that we can see in urine as well. Some things we do not want to see in the urine. Glucose, okay, that's something that we do not want to see in the urine. That's a test that we will take, right, to measure proteins or proteins in the urine. That wouldn't be good. That means there's something wrong with the kidneys. The blood proteins got out into the filter. We want to keep our glucose, so we don't want a lot of glucose in the urine as well. That can indicate some problem with filtration, or it could mean that we just had a really sugary meal, some glucose leaked out. We have a lot of it. So our kidneys, the major organs that are allowing for this filtering of the blood and then excretion of the filtrate, which is urine. So they are producing urine. They take the filtrate out of the blood. Again, that's the liquid part of the blood, minus the blood cells, minus the blood proteins. Maybe some ions in there and other nitrogenous components. That filtrate gets changed and changed and changed as it's traveling through the kidney into urine. And the urine will leave the kidneys through the ureters that will deliver the urine into the bladder, and then we would eliminate the urine out of the urethra. So our urinary system, as the blood is being filtered at the kidneys, all of that in our digestive tract, all of that, the nutrients that we absorb, if it gets filtered out, we quickly pull that back and put it back into the blood. And we do all that digestion, but not some work, right? Going through the digestive tract. So we don't want to lose any important nutrients. But if we happen to filter something out at the kidneys will quickly put all that good nutrients right back into the blood. And we'll only eliminate what we want to. Leaving the kidneys, traveling through the ureters, down to the urinary bladder, and out of here. Let's look at the kidney itself. Again, filtering the blood, getting rid of waste, getting rid of toxins, regulating our blood volume again, because blood volume, that's blood pressure. The kidneys not only participate in filtering the blood, but they participate in the endocrine system. They secrete a hormone, renin, that will help to regulate blood pressure. Blood pressure is low. Kidneys secrete renin. They also secrete erythropoietin. This stimulates the bone marrow to make more red blood cells when necessary. And they participate in activating vitamin D, helping to produce glucose as well. When we are fasting, they help with glucogenesis. Our urinary system. Structures on top of the kidneys are the adrenal glands, not a part of the kidneys, their own separate organs, a part of our endocrine system. Again, the kidneys, just like we saw the lungs, they have a hilum. Region there where blood vessels are entering, ureters are exiting. Those ureters will head down, deliver urine into the bladder, and we'll excrete that out of the urethra. nice cross section through the body where we can see the kidneys. So we're gonna take a look. Posterior view. Those adrenal glands again are just part of the endocrine system, not a part of the kidneys, but important part of the endocrine system. They secrete neurotransmitters, okay, epinephrine, norepinephrine, our fight or flight neurotransmitters, okay? Adrenaline. Okay, so that the hilum, the renal hilum, okay, is that structure, that concave area here. We have blood vessels entering and exiting, as well as the ureters exiting.
So are they on their lower back? Can they be somewhere else?
It shouldn't be.
What if it is? Was it, I mean, you wouldn't know the function, right?
If everything is intact, I mean, where would they be?
Well, my son had one right there, in the garage, in his lower abdomen. They found it and they took out his appendix, but they
They
never.
function fine, if
They
everything
don't
is.
know, because he was, 6 or 8 when they did it. They said they would help with you, you know, when they put the dye in, is it the,
Yeah.
CT or yeah? And they said he was little and it could cause other, you know,
Yeah.
worse damage,
Mhm.
so they stayed with him.
But
But I mean, he's
they would have told
healthy,
you if,
fine.
right, if he was unhealthy, right? So
Yeah, they're
I
like,
think as long as everything is functioning fine.
That's weird, though, right? I mean, he's different. He's different. He's different.
What?
So you said the kidneys
Yes,
start
sir.
with, or fat, basically, with that,
So,
I'll just do it later. This is
Oh,
what-
yeah, yeah. That's our sugar source. Our energy source that changes, is changed into ATP, our energy.
Oh, so the kidneys of that? Wow. What
When
about
we are
that?
fasting.
When we're fasting.
Yes.
Is that what creates us to go into ketosis?
So, that's good, we're gonna talk about that.
Okay.
Yeah. Ketoacidosis, yeah, part of that. Okey dokey. All right, so we have some tissues that help to support the kidneys. There's a fascia, connective fibers connective tissue that helps to anchor the kidneys, okay, to other structures in the body. It has a fatty cushion, and then it has this fibrous capsule. And below that, get even deeper, you're gonna head into the cortex. Now we're getting to a region where we're gonna find very important structures that help with the filtration of the blood, okay, at the renal cortex. And then we go even deeper and we're at the medulla, okay? The words that are common with organs, cortex, and medulla. The medulla here, which is the more central region of the kidneys, are arranged in these pyramid structures called renal pyramids. You'll see how the renal cortex kind of surrounds each of the renal pyramids. And when the cortex dips in between two pyramids, they get a different name. It becomes renal columns. The cortex becomes renal columns in between two pyramids. Okay, so we'll see that. So here we're looking at the capsule. Okay, this fibrous tissue that anchors the kidneys to the rest of the body, or the structures around it. And then we're in the kidneys here. The first layer, the most superficial layer is the cortex. And then the deeper layer is the medulla. And the medulla is organized as these pyramids, renal pyramids. So here's the cortex. The capsule is the outermost layer, fibrous layer, then the cortex, and then the medulla, deeper regions arranged in these pyramid-shaped structures, renal pyramids. And as the cortex dips between neighboring pyramids, the cortex is called a renal column. So here's a renal column, here's a renal column. So when we see these in the model, we'll see the renal pyramids, which is the medulla, and the cortex. When the cortex dips between two renal pyramids. We call that the renal column. So the pyramid, the very tip of it, that bottom tip, is called, you see this word so much, papilla, are you? I'm tired of it, I've seen it a lot. That's the very tip of a renal pyramid. And this is where urine was gonna drip down into what eventually will lead into the ureters and out the kidneys. Okay, that sink that's going to receive the filtrate, the urine, is called a minor calyx. We'll see that. Those pyramids of medulla with the cortex surrounding it, if you take a pyramid with the cortex surrounding it, we call all of that a lobe. Okay. And then we'll see the renal pelvis and how that leads us to the ureter. So on our model, we're gonna call this the renal lobe. Now, it should have some renal column on both sides. It doesn't, but we're gonna let this, because here's cortex. And then here's the renal pyramid, or the medulla. You can see there's a lot of structures in here, and these structures are the units that will filter the blood, the nephrons. When the blood is filtered, we get the filtrate at the very tip again of that renal pyramid, that papilla. The filtrate will drip into a sink, and that sink is the minor calyx. And it's gonna catch the filtrate or the urine, out of the urine, and deliver the urine into the major calyxes. So minor calyxes will merge to form major calyxes. And then urine will flow from a major calyx into the renal pelvis. So major calyxes will merge to form the renal pelvis. And from the renal pelvis, we enter into the ureter. So here's a flow. From here, arrow to there, arrow to there, arrow to there, from the renal pyramid into the minor calyx, into the major calyx, into the renal pelvis, into the ureter, into the bladder, out of the urethra. And there's more before renal pyramid. Add to that list. Okay, see those things. So here's a renal pyramid. This is how the medulla is organized in these pyramidal shapes. The very tips or papilla of each pyramid is right at a minor calyx that acts as a sink, collecting the urine as it drips from the tip or papilla of the pyramid. You can take two or a few minor calyxes, and they will merge to form a major calyx. And then the major calyxes, minor calyxes, merging. So each one is associated with a papilla or tip of a renal pyramid, catching the urine as it's dripping from the tip or papilla of the pyramids. Those minor calyxes merge to form a major calyx. And those major calyxes will merge to form the renal pelvis. And this renal pelvis will take it down into the ureter. So there again, a renal pyramid, a minor calyx, merging into a major calyx, and then the renal pelvis, and then into the ureter. Now, the point of the kidney is to filter the blood. So we need to get the blood into the kidney, right? Think about how blood gets to the kidneys. We have our renal artery bringing in blood. Eventually, the blood is gonna go through its process and then leave the kidney through the renal vein. But you have to do some traveling. And along that travel, you have to get filtered. And then after being filtered, we can exit the kidney through the renal veins. But then that filtrate has its own path that it has to travel through. Talk about that. And that would be the tubule system through which the filtrate has to travel. So we enter into the kidneys through the renal arteries, and you all can follow along on the slides, you have them on campus. So feel free to take your notes on your slides. You're entering in the renal artery. And that renal artery, as you can see, is gonna start to branch. It will branch into segmental arteries. The segmental arteries kind of lie on the major calyx. And then those will branch into lobar arteries, kind of lie on the minor calyx. We're trying to get here. This is where we're trying to get the blood to. Because this is where we're gonna find the structures that are gonna filter the blood. So we have to go from here, and we're trying to get the blood all the way here. Now we're at the lobar arteries. From there, we'll travel along the interlobar arteries that are lying on those renal columns. And then we're going to arc, or over, the tops of the pyramids as the arcuate arteries. And then those are gonna take us into these small arteries that are radiating along the cortex. And those are the interlobular, different from interlobar, the interlobular, or the cortical radiating over the cortex arteries. And now we're getting closer and closer to the structures that are gonna filter the blood. So, to remember, real students love interesting anatomy and structures, with how we're gonna remember renal arteries, taking us into the segmental arteries, leading us into the lobar arteries, taking us into the usate. Okay. Once we've been filtered, these interlobular arteries are gonna take us closer and finally into the structures that it's gonna filter us. There's the blood, we're traveling. We travel through these, we're trying to get to the place we're gonna get filtered, we'll see where that happens. After we're filtered, we're going to leave the kidney, nice and clean. And we're gonna then enter to the venous side. And we're gonna leave along the interlobular veins. That will then deliver us or drain us into the arcuate veins that arc over the tops of the pyramids. That will then drain us into the interlobar vein. And then finally, out of the renal vein. And back, now we're on the venous system, so where are we headed to? The inferior vena cava, taking us into the going into the right atrium. Okay, it's all connected, yes. Okay. All right. To remember the venous flow, leaving the kidneys, you say, taking us into the, and then draining us into the, and finally we're leaving the kidneys in the, that's right, making our way back to the heart, right, to deliver this deoxygenated blood to the lungs, okay, do I need to, got it, right? Okay, good. Now, let's talk about this. That's right. Get us closer and closer to the lungs for gas exchange, right? Okay, the nephron. This is the structure that we're trying to get to, okay? From those interlobular arteries, we need to get into the nephron. The nephron is going to filter us as we're blood traveling through the renal system. It is the structural and functional unit of the kidney. And we have about 1 million nephrons per kidney, working on filtering our blood. The nephron consists of the renal corpuscle and the renal tubule system. This is how filtrate will travel. So blood is being filtered here, coming in through that renal artery, making its way to the renal corpuscle. Gets filtered, and then leaves on the venous side to go out of that renal vein. But as it's being filtered in the renal corpuscle, that filtrate is being formed and collected. And that filtrate is traveling in a whole other system, the tubular system of the nephron. And it's the filtrate that will become urine after it goes through all the changes it goes through, with things being pulled out of it, because we want to keep that, and things being secreted into the filtrate, because we want to get rid of that. And that's gonna change the original filtrate formed here as the blood has been filtered into something totally different, what we call urine. It's gonna look different from the liquid that gets pushed out of the blood originally. At the renal corpuscle, every nephron has a renal corpuscle that's composed of two parts, the glomerulus and the glomerular capsule, also called Bowman's capsule. As we enter into, so this is here, we're going in from that interlobular artery. We're then gonna enter into an afferent arteriole, and that's gonna take us into the glomerulus. The glomerulus is a bunch of, a ball, tuft of capillaries, our thinnest wall blood vessels. We want thin wall blood vessels here, because we're trying to push liquid out of the walls. So we need those walls to be thin. So here we're dealing with capillaries. But not only capillaries, fenestrated capillaries. What does that mean? They're whole, porous, lots of holes in them, because we're trying to filter and push fluid out of the capillaries. That fluid that's getting pushed out of the blood, that's traveling through these capillaries are called the glomerulus. As that fluid gets pushed out into the glomerular capsule that's capturing that filtrate, the blood continues to travel as it's being filtered, and finally will leave out of what's called the efferent, efferent, exit, that's how I remember. A comes in, afferent arteriole, afferent, to affect. I'm about to go in here and I'm gonna affect this glomerulus, afferent. That's how I remember it. You remember it your way. And then when I'm done being filtered, I'm ready to exit. E, efferent arteriole, takes us now onto the venous side. Okay? And the filtrate is captured in the glomerular capsule. Bowman's capsule, glomerular capsule, interchangeable. Histologically, here is the glomerulus. Every nephron has a glomerulus. Or a renal corpuscle is the glomerulus plus the glomerular capsule. The blood enters the glomerulus, gets filtered as it's traveling through the glomerulus. The filtrate gets collected in this capsular space, okay, that's made by the glomerular capsule cupping the glomerulus, okay? It's an enclosed ball, this renal corpuscle. There it is again, renal corpuscle. With the glomerulus and the glomerular capsule, also called Bowman's capsule, making this capsular space that filtrate is being filtered into, so that it can head down into that tubule system of the nephron. Bowman's capsule, parietal and visceral layers, we've heard that. See this everywhere, everything has a parietal layer and a visceral layer. Parietal layer and a visceral layer. The parietal layer is here. This is the parietal layer of Bowman's capsule. Like every parietal layer, it folds back upon itself and lines the structure that it's covering. Here, it lines the capillaries of the glomerulus. And it lines it as a whole other set of cells that sit on top of the blood vessels, the capillaries here. We're gonna see those cells. So the parietal layer is some simple squamous epithelium. Thin, long, simple squamous epithelium of the glomerular capsule will fold back upon itself and line the capillaries of the glomerulus. That is the visceral layer. And the cells of the visceral layer get a special name. They're called podocytes. You're gonna see this on the urinary tract model, the podocytes. The podocytes are special because they take their plasma membrane and kind of make these processes that then interlink to make these sifting structures. And that aids in the filtration of the blood. Okay? So these foot processes help to filter the blood, okay, as it's traveling through the glomerulus. Those filters act as the slits, filtration slits that are created by the foot processes of the podocytes that are laying on top of the capillaries of the glomerulus. They act as sieves. And so if anything big gets pushed out of the blood, okay, it has to get sieved by these filtration slits. These filtration slits help to keep back anything that's big that tried to get out of the blood. And then those things will be like proteins that should not leave the blood. Those things will get reabsorbed back into the blood right here at the glomerulus. All right, let's check out how that looks. Okay, so here's the parietal layer of the Bowman's capsule, also called the glomerular capsule. It makes the capsule, and then it folds back upon itself and lines the capillaries of the glomerulus. Okay. These are fenestrated, porous holes in it, the walls of this capillary. The parietal layer folds back upon itself and becomes the visceral layer. And the cells of the visceral layer get a special name, podocytes. And they use their plasma membranes to make these foot processes that make these slits that aid in filtering the blood. So as blood is traveling through this capillary, here's the wall of the capillary. There's a little basement membrane in between both that aids in the passage of ions and so forth. But as blood is traveling through the capillaries, if large things get pushed out, these filtration slits help to allow small things to keep getting pushed out, but hold back large things that would get reabsorbed back into the blood. So here is the renal corpuscle. Renal corpuscle. Here's that afferent arteriole that leads us into the glomerulus. Here it's just all stretched out. It's not all bundled up. Okay, like it is normal. We stretched it out. We're coming in through the afferent arteriole of blood, traveling through the capillaries that are fenestrated, so we're getting filtered. Liquid and small ions are getting pushed out, okay, into the capsular space. Here's Bowman's capsule, the parietal layer. It folds back upon itself and lines the walls of the capillaries of the glomerulus now as the visceral layer. And these cells get a special name, podocytes. And they are aiding in the filtration. They will not let anything large get out. If it got out of the blood, they won't let anything large get past into the capsular space. So only liquid water and small ions like glucose can get pushed out into the capsular space. The blood continues to travel through the glomerulus as it's being filtered, and then exits the glomerulus through the efferent arteriole and heads towards that renal vein. The filtrate has been collected here in the capsular space, and that's gonna make a whole other trip into the tubule system of the nephron. And it's in that tubule system that the filtrate will be changed into urine. This is our model. There's the glomerulus. There's the glomerular capsule. Together, these two are called the renal corpuscle. There's that afferent arteriole taking us into the glomerulus. Here are the podocytes lying on top of the glomerulus here, they're removed, so you can see the actual capillaries of the glomerulus that are porous for filtration. We enter into that afferent arteriole, travel through the glomerulus, filtered as we're traveling, and then we exit out the efferent arteriole. The filtrate is collected in Bowman's capsule. And then that filtrate heads into the tubular system of the nephron. That membrane that's made with the capillary and the podocytes on top of them has a special name, the filtration membrane. It's the membrane between the walls of the capillaries of the glomerulus and the podocytes sitting on top of it, so that any large macromolecules will get stuck in those filtration slits made by the podocytes, and they can't leave and get out to the filtrate. And so they will reabsorb back into the blood, into the capillary. So this filtration membrane only allows molecules that are smaller than 3 nanometers to pass through. So now we're talking about very small things, glucose, which is one sugar, right? One single molecule sugar. Amino acids, water can head out. Blood proteins are too big. They will stay back and get reabsorbed back into the bloodstream. So here's a good depiction of the filtration membrane. The fenestrated capillaries of the glomerulus, with the podocytes on top of them, making these filtration slits with their plasma membrane. A nice electron micrograph of that. Nice depiction of the wall of the capillary of the fenestrated wall of the capillaries of the glomerulus. The filtration slits made by the plasma membranes or the foot processing of the podocytes, with a little basement membrane in between them that allows ions to easily pass through. Once we've been filtered, and now we're filtrate that's been collected here in the capsular space. It's time to head as filtrate into the tubule system, starting at the first part of the tubule system, called the proximal, closest to the glomerulus, convoluted tubule. From the proximal convoluted tubule, we then head down the nephron loop, also called the loop of Henle. This is where filtrate is changed into urine. All of it is where it's getting changed into urine. At the proximal convoluted tubule, that's when everything that got pushed out of the blood that we want to keep quickly gets put back into the blood. So glucose, we want to keep that. So any got pushed out of the glomerulus, right there at the proximal convoluted tubule, glucose will be put right back into the bloodstream. So if we're now in this tubular system of the nephron, and things that have gotten pushed out of the blood are quickly put back into the blood, what needs to be near this tubular system? If we want to quickly put things back into the blood, what needs to be right there at the tubular system so that can happen? What would you say? Some kind of capillaries. They can be right there too. We're gonna have a whole other set of capillaries ready to receive anything that needs to quickly get put back into the blood. Okay? Let's see those are coming. First, let's think about the filtrate. Travels through the proximal convoluted tubule, closest to the renal corpuscle. Down the loop of Henle, it has a descending limb, so we go down the loop of Henle, and then up its ascending limb into the distal convoluted tubule. That's farthest away from the renal corpuscle. And you can tell the difference between the proximal and distal. The proximal in my histological sections always looks fuzzy. Okay, so histologically, the lumens are fuzzy of the proximal convoluted tubules as compared to the distal convoluted tubules. Here's the glomerulus. distal convoluted, nice and clear, proximal, fuzzy one. The proximal convoluted tubule, right there when we leave the Bowman's capsule, right when we've been filtered out, we go into the renal, tubule system. This is right there is where water gets pulled right out. Whatever water got pushed out, not all of it gets pulled back in. We're gonna quickly take some of it, depending on how thirsty we are. Okay? We're gonna take some of that water back and put it back into the blood. Okay? Organic nutrient, glucose. We're gonna quickly grab any glucose that got pushed out into the filtrate, because we need that to make ATP. And we're gonna quickly put that back into the blood. Again, we need a whole other capillary system here that's gonna be able to take things back into the blood as we're pushing them, filtering them back into the bloodstream. At the same time, there's things in the blood still that we might wanna put into the filtrate that maybe didn't get filtered at the glomerulus. That's secretion. So at the same time, while we are reabsorbing things out of the filtrate back into the blood, we are also secreting things out of the blood into the filtrate as it's traveling. So we're continuously reabsorbing what we want to keep and secreting what we want to get rid of. As this absorption and secretion is happening, that filtrate is being changed and concentrated into urine. Once we leave the proximal convoluted tubule, we head down the nephron loop, also called the Dubochin loop, the hairpin, down and up. We go down the descending limb and up the ascending limb. Different things are happening. At the descending limb, water is being reabsorbed back into the bloodstream. At the ascending limb, we have salt, sodium and chloride can be absorbed back into the bloodstream. So different things happen along the nephron loop. Different ions are focused on along the entire tubule system here. Water is focused on in some regions and not in others. When we get to the distal convoluted tubule, we won't be worried about something that we've already collected as the proximal convoluted tubule. Okay? So at the distal convoluted tubule, once we leave the loop of Henle, we're in the distal convoluted tubule. Here we're secreting things that we don't want out of the blood and putting it into the filtrate. Here we are reabsorbing salts, calciums, but we're not worried about reabsorbing water back into the bloodstream. We did a lot of that already. And so as the filtrate is traveling through this tubular system of the nephron, different things are being focused on as far as what's going to continuously be reabsorbed out of the filtrate because we want to keep it. Or depending on the body's needs, we need that, so we need to grab it back out of the filtrate. We don't want to urinate that out. We want to keep that. Or we're secreting things out of the blood. We gotta get rid of this. We want to urinate this waste out. So know the difference between reabsorbing out of the filtrate back into the blood or secreting out of the blood into the filtrate to get rid of it. So let's look at that system. So there's the glomerulus, the renal corpuscle. The glomerulus encased in the glomerular capsule. There's that arcuate artery. Okay, that's on top of a renal pyramid. It's not there, but the renal pyramid would be here. Renal pyramid would be here. That arcuate artery leads us into the interlobular artery. It takes us into the afferent arteriole. Now we enter into the glomerulus. Traveling through that capillary system, the glomerulus being filtered. And then, once we're done being filtered, we leave out of the efferent arteriole. And then this delivers us into the interlobular vein. We go out the kidney eventually. But that filtratus here has been captured in that capsular space, and it enters into the tubule system of the nephron, starting at the proximal convoluted, all twisted up, that's what convoluted means, right? Convolutions. The proximal, nearest little renal corpus, proximal convoluted tubule. Here, we're quickly reabsorbing water, quickly reabsorbing glucose, things we want to keep back into the bloodstream. So that means there has to be another capillary system nearby. There is, it's just not drawn here, but we're gonna see it. In order to take stuff out of the filtrate in this tubule system, proximal convoluted tubule, and put it back into the bloodstream, because we want to keep it. As the filtrate is traveling, the proximal convoluted tubule, it heads down into the loop of Henle, down the descending limb, where certain things are focused on, more water being reabsorbed, salts, and then we travel up the ascending limb, what other things are being focused on? Other ions are being focused on, being absorbed back into the bloodstream or secreted out of the blood into the filtrate. And the filtrate keeps traveling. Now we're in the distal convoluted tubule. Convolutions. And finally, every nephron will deliver from their distal convoluted tubules filtrate into a collecting duct. So a collecting duct is receiving filtrate from many different nephrons. At our model, we're going to see the nephrons. Here's a renal corpuscle with a glomerulus encased in a glomerular capsule. Renal corpuscle, renal corpuscle, renal corpuscle, renal corpuscles. Here is that interlobular artery with all of these afferent arterioles taking us into these different glomeruli inside of these renal corpuscles. Blood is filtered. The filtrate gets captured by the glomerular capsule in that capsular space. And now it enters into the proximal convoluted tubule. I know this one is labeled. And then down the loop of Henle. The loop of Henle dips all the way down into the medulla. That means something. This is at the loop of Henle that urine is gonna be concentrated. It goes deeper and deeper down into the medulla as it's traveling in the loop of Henle that dips down deep into the medulla. And then finally up the ascending limb of the loop of Henle and through the distal convoluted tubule and delivered into a collecting duct. This collecting duct was then gonna drip that filtrate down into a minor calyx at the very papilla, the tip of this pyramid. And then we're gonna start getting rid of that urine out of the ureter, bladder, urethra. So there's a loop of Henle that dips down into the medulla. Here is a longer loop of Henle. These are the special ones that concentrate urine. But as that filtrate is traveling, we're constantly secreting from the filtrate back into the bloodstream, or secreting from the bloodstream into the filtrate as it's traveling, or reabsorbing out of the filtrate and back into the bloodstream. Things that we want to keep. Okay, so that's blood supply. Then we came in through that renal artery, then we went into the segmental arteries, and then into the lobar arteries. I can see myself asking you to explain the blood flow. Renal artery, arrow, segmental artery, arrow, lobar artery, arrow, interlobular artery, arrow, argular artery, arrow, interlobular arteries, arrow, afferent arterial, arrow, glomerulus. Now we're in the glomerulus, we're getting filtered. That filtrate is entering into the capsular space. It will have its own travel in the tubule system. Proximal convoluted tubule, arrow, descending loop of Henle, arrow, ascending loop of Henle, arrow, distal convoluted. Following me? Follow me, med school? That's a big.
Yeah, all that stuff goes from that word.
After we get filtered, we're ready to leave the glomerulus through the efferent arteriole. But the efferent arteriole, before we get to the venous system, the efferent arteriole is gonna take us into the other capillary systems that I said have to be here in order to still secrete out of the blood and into the filtrate or reabsorb from the filtrate into the blood. And those two different capillary systems are the peritubular capillaries and the vasorecta. Two additional capillary systems there in order to receive things that are being pushed out of the filtrate and into the blood, or to push things out of the blood into the filtrate as the filtrators travel. And then finally, we enter into the venous side of things. We're defining to the renal vein and into that inferior vena cava, making our way back to the heart. So as we travel all the way from the aorta, making our way to the renal artery, all through those arteries we talked about, into that afferent arteriole, then the glomerulus, then out of the glomerulus to the efferent arteriole. We can either go into the peritubular capillary system or the vasorecta capillaries. Once we travel through there, where we're either still filtering things out of the blood, secreting things out of the blood into the filtrate, or we are reabsorbing things out of the filtrate back into the blood. Once we've done all that, then we leave out the venous side. And so how things really look over here first is like this. There's the tubule system, all encased with a whole other capillary system, ready to receive what's being filtered out of the filtrate. Still, as it's traveling, stuff is still getting pushed out of the filtrate that we want to keep and will reabsorb back into the blood. And still, as the blood is traveling through this capillary system, there's things that we still want to get rid of that get secreted out of the blood, travel through the interstitial space, the tissue, and into the filtrate. So right there at the glomerulus, here, the renal corpuscle. Once we've been filtered, the blood travels through that efferent arteriole. Efferent arteriole. That leads us into the peritubular capillaries. Let's go back one. When it comes to our nephrons, we have two types of nephrons. We have the cortical nephrons. The cortical nephrons, the majority of our nephrons are the cortical nephrons, have short loops of Henle. They don't dip deep down into the medulla. A little bit, but not deep down into the medulla. Okay? So the majority of the cortical nephrons are entirely in the cortex. They have short loops of Henle that dip a little bit down into the medulla, but not far. If you're a cortical nephron, your efferent arteriole delivers blood into the peritubular capillaries that would eventually take you into the renal side or the venous side. So this would be a cortical nephron. I'm gonna call this a cortical nephron, where its efferent arteriole delivers blood into the peritubular capillary system. That's right there at the convoluted or the proximal convoluted tubules and surrounding the distal convoluted tubule. Ready to pick up things being pushed out of the filtrate and ready to secrete things into the filtrate. Out of the blood, secrete out of the blood into the filtrate, or reabsorb out of the filtrate back into the blood. That's if you're a cortical nephron. If you are a juxtamedullary nephron, you have a long loop of Henle that is deep down into the medulla. Your efferent arteriole will deliver blood into some peritubular capillaries, but also the vasorecta. The vasorecta capillary system surrounds that long loop of Henle. So here, if you are a juxtamedullary nephron, you have a long loop of Henle that moves deep down into the medulla. And you are surrounded by some peritubular capillaries, but also the vasorecta, a whole other capillary system that surrounds that deep dipping loop of Henle. And it is helping to concentrate the urine as it's traveling through this long loop of Henle. It's receiving whatever is being pushed out of the filtrate and being absorbed into the bloodstream here, or it is secreting whatever we want to get rid of in the blood out into the filtrate as it's traveling. So again, we're continuously filtering and secreting, or reabsorbing out of the filtrate and secreting into the filtrate. And that's how the blood would originally get pushed out of the glomerulus here. It's totally different when we get over here. Since we've done a lot of pushing things into it and pulling things out of it, including water, so we're concentrating this. Okay, so when your urine is really yellow, that means your kidneys have pulled out a lot of water because you're thirsty. When your urine is nice and clear, that means we're not worried about pulling out a lot of water. You're nice and hydrated, so we're gonna let all that water pass. So on our model, we're gonna see an example of a cortical nephron and a juxtamedullary nephron. The loops of Henle. Every nephron is going to deliver its filtrate into the collecting duct. The collecting duct runs all the way down to the papilla of the medullary pyramid. And it's from here that that final filtrate is dripped down into a minor calyx, that first little sink that's capturing the filtrate as it's dripping out of the collecting duct at the papilla of the nephron. So, the three capillary beds associated with the nephron is the glomerulus, where you first get filtered. The peritubular capillaries, if you're a cortical nephron, that's the capillary system surrounding your tubule, or the vasorecta, and some peritubular capillaries if you're a juxtamedullary nephron. So that glomerulus job is filtration. That makes it different from the peritubular capillaries and the vasorecta capillaries. The glomerulus is just all about filtering. Where again, the blood is entered in through that afferent arteriole. It filters the blood, and it allows the blood to then leave through the efferent arteriole. And through the efferent arteriole, that blood would enter into those other two capillary systems. The glomerulus, in the blood glomerulus, blood pressure is high because that afferent arteriole coming into the glomerulus is a larger diameter compared to the efferent arteriole leaving the glomerulus is a smaller diameter. So that's bringing a lot of blood and only making a small exit. So the blood builds up. More volume, more pressure against the walls of those blood vessels. So in the glomerulus, you have high blood pressure, but that aids in pushing the blood through the holy walls of those capillaries. That pressure helps to filter. So we want that high blood pressure, okay? Once we enter into the peritubular capillaries, if you're a cortical nephron or a juxtamedullary nephron, you have the peritubular capillaries associated with you. You will leave your efferent arteriole and you enter into either one of these, or into the peritubular capillaries. Now we have low blood pressure. And we want that to be low blood pressure. We want the blood to travel slow. We want it to get time for absorption. We're trying to pull the water out of the filtrate and back into the blood. So we're gonna let the blood take its time so that it can receive whatever needs to get back into it real quick. Water, glucose. Again, the vasa recta is gonna be surrounding the long loops of Henle of the juxtamedullary nephrons. And the way that loop of Henle is constructed, it goes down and up. It's gonna aid in the concentration of urine. And these juxtamedullary nephrons help with concentrating the urine. They allow for exchange of water and other molecules, okay? That as the filtrate is traveling, the deeper it goes down into that medullary pyramid, the more concentrated it's going to get. Okay? And so, how it really looks is like this. Cortical nephron has the peritubular capillaries associated with it. The juxtamedullary nephrons can have some peritubular, but definitely vasa recta, surrounding its long loop of Henle. And it's the long loop of Henle here and the vasa recta that help to concentrate urine. Okay. So we also have what's called the juxtamedullary complex. At every nephron, you have this complex. And this complex is just a group of cells that are important. We have a group of cells called the macula densa. We have a group of cells called the granular cells. And we have a group of cells called the extraglomerular sagittal cells. These three populations of cells make up what's called the juxtaglomerular complex. And these cells are important because they help to regulate blood pressure. They help to regulate the rate of flow of filtrate. They're sensing things in order to regulate the flow of filtrate. So here we are. Here's the glomerular, the renal corpuscle, made of the glomerulus and the glomerular capsule. There's that afferent arteriole taking us into the glomerulus. And here's the efferent arteriole leading us out into the peritubular capillaries or the vasa recta. Here is that juxtamedular apparatus or complex made up of three populations of cells. The macula densa. These are just 5-6 cells in the wall of the distal convoluted tubule that makes its way right up against the glomerulus. And then a population of cells, the granular cells, in the wall of the afferent arteriole. And then in between these two populations of cells are these guys. These are the extraglomerular sagittal cells. They help to control the dilation of the glomerulus. By expanding themselves, they affect the constriction and by dilation of the glomerulus. And that is the juxtamedullary complex or apparatus. It's these three populations of cells that are important. The macula densa cells in the walls of the distal convoluted tubules are chemoreceptors, and they're sensing the salt concentration of the filtrate as it's traveling through the distal convoluted tubule. If the salt concentration is high, that will cause a slowdown of the flow of the filtrate through that afferent arteriole. This is coming into the glomerulus. Slow that down to increase the time the blood is being filtered at the glomerulus and start getting some of that salt out of the blood. So that's their job. We'll see those on the model. There's the distal convoluted tubule, macula densa, are these 5-6 cells that act as chemoreceptors and they are sensing the salt concentration of the filtrate traveling through here. If that salt concentration is high, they will cause the slowdown of the blood here, coming in the afferent arteriole. Slow it down. So now you slow down how long it takes for the blood to be filtered, so that you give it time for salt to be filtered out. We can see macula densa in histology pictures. Here is an electron micrograph of the cells of the macula densa. Chemoreceptors sensing salt in the filtrate. The granular cells, or also called the juxtaglomerular cells. These are cells in the smooth muscle cells in the afferent arteriole. These are mechanoreceptors receptors. They are sensing blood pressure in the afferent arteriole. And they will trigger the kidney to do its endocrine job, to release renin in order to regulate blood pressure as a part of the renin, angiotensin, aldosterone, hormone system. So when you look at the endocrine system, you should see renin as a part of this whole process. And this renin is secreted when blood pressure is low, in order to raise the blood pressure. So these granular cells are sensing blood pressure. And if blood pressure is low, then it triggers the kidneys to release renin in order to increase blood pressure. So here's the afferent arteriole. Here are the cells, the granular cells in the wall of the afferent arteriole. If blood pressure is low, traveling through that afferent arteriole, these cells will cause the kidney to secrete renin and increase the blood pressure. And then those extraglomerular mesangial cells are cells that lie in between the macula densa cells and the granular cells. And they have a lot of gap junctions. You know what that's for? To interconnect those cells and allow them to share ions. They allow ions to pass between the macula densa cells and the granular cells. Here we are again, a juxtaglomerular apparatus or complex. These three populations of cells sensing salt and slowing down the rate of filtering so that salt can get filtered if it's high content being sensed in the filtrate, or sensing blood pressure that will cause blood pressure to be raised if it's low. And then the granular cells will allow these two populations to communicate. They also affect the constriction and dilation of the glomerulus. Okay, filtrate is flowing. Gets into that renal tubule, the proximal convoluted tubule, down the loop of Henle, up the loop of Henle, okay, and then the distal convoluted tubule. From there into the collecting duct, captured, then drifting down into the minor calyx, that merges into a major calyx, that merges into, I left one part out here, the renal pelvis, that then leads us into the ureter, then down into the bladder, and out the urethra. So those collecting ducts are receiving filtrate from many different nephrons. And then they will deliver that into the minor calyxes at the tips of every medullary pyramid. We'll have a minor calyx ready to receive the filtrate as it drips down from the collecting duct. So glomeruli are here in the cortex, and here's a collecting duct traveling down through the medulla to the very tip or papilla of the pyramid. Here's a minor calyx right there at the pyramid, the papilla, ready to capture the filtrate as it's dripping out of the collecting duct. So the minor calyxes will calyx drain into the minor calyxes. These are collecting that's traveling through them. And that will then drain into the major calyx, and then drain into the renal pelvis, and then drain out of the ureter. And then we are headed towards the bladder. And the ureters are going to enter the bladder at a kind of a triangular shape region of the bladder called the trigone, inside of the bladder. And the triangular shape is called the trigone. The ureters, their openings are there at the trigone of the bladder. So here we are, see a model like this. Here are the ureters that will enter into the bladder at the structure inside of the bladder called the trigone, where we have the two urinal openings and then the pathway to the urethra. And now we're thinking of the histology. Now we're thinking of transitional epithelium. Remember that? All interconnected. That transitional epithelium is going to expand or relax depending on if the bladder is full or empty. The ureters have some muscle layers surrounding them, some smooth muscle. Always smooth muscle associated with every organ except for the heart. What kind of muscle there? Heart. Yeah. And then there's an outer layer of adventitious, a fibrous connective tissue that anchors the ureters to other structures in the body so they're just not moving around. Histologically, we can look inside of the ureter. We can see its muscular wall. We can see its lumen, its inner layer, which is a mucosa. So it secretes mucus, but it's also consistent of that transitional epithelium. It's adventitia that anchors it to surrounding body parts. And the ureters deliver that urine into the bladder at the trigone. That bladder is also consisting of transitional epithelium. There's also the detrusor muscle here, this thick muscle that's gonna be relaxed to allow the bladder to store urine, but then will contract in order to cause the bladder to release urine. So here is the trigone again, the openings of the ureters into the bladder, and then leading into the exit of the urethra. And the urethra is just a muscular tube that's helping to drain the bladder. Still some transitional epithelium that's gonna change into some stratified squamous epithelium. Right, we need some protection. That's that protective tissue. As we lead through the urethra. There's a difference between the male urethra and the female urethra. The male urethra has three parts. The prostatic urethra that runs through the prostate gland. Into the membranous urethra, very short region of the urethra. Continuous with the spongy urethra that runs through the penis. So we'll see the urethra, the male urethra, on the male reproductive system, the prostatic urethra, all connected. The membranous urethra, very short span of the urethra, into the penile urethra, out of the penis, compared to the female urethra. Okay. We will see on the female reproductive system, leaving the bladder out of the urethra. Transitional epithelium, don't forget it. Very important and helpful in helping us to expand our bladders to allow them to fill up with urine, or to relax and help to push the urine out. Forget what a relaxed urethra or bladder looks like compared to a distended or full bladder. The process of urination or voiding is micturition. And again, the detrusor muscle around the bladder contracts, and there's a sphincter that we don't control, the internal urethral sphincter. It's controlled by, involuntarily by the autonomic nervous system, but then we have one that we can control. Okay? The external urethral sphincter. And you're done.